The science of inheritance is called genetics. Heredity is a biological process where a child or an offspring inherits certain genes from their mother and father. All children inherit genes from their biological parents. The genes express distinct attributes. The attribute might be physical characteristics like skin and hair color, body shape and size. Regrettably, some genes bear the danger of indefinite diseases and disorders that might pass from the parents to the offspring. Lean body mass is hereditary trait which can predict various types of health issues, in particular orthopedic problems such as sarcopenia and osteoporosis. Sarcopenia is a progressive decrease in skeletal muscle mass and function caused by aging. It is also associated with common diseases such as cardiovascular disease. Osteoporosis is a disease with a forceful genetic factor. It is characterized by deterioration of bone tissue, lower bone mass, and deterioration of bone tissue, and susceptibility to fractures. In addition, the loss of function and impairment of skeletal muscle is a common problem that affects a lot of people around the world, specifically the older population. Heritability is estimated to range from 52 to 84%. Traditionally, the association analysis is focused on single nucleotide polymorphism, also known as SNPs. The assessment of SNPs variation has been conducive, proving a lot of common variants to be linked to diseases, i.e. osteoporosis. Recent research have shown another type of genomic variation, a copy number variants, CNVs, to be an important in prompting diseases also, and very feasibly to be prevalent in the population. The study tried to look at the sequence variants affiliated with lean body mass by Genome-wide association study of copy number variants, CNVs. In this study, they genotyped genome-wide CNVs of 1,627 participants from Chinese population. The participants were recruited from the study, which aimed at seeking the genes that are responsible for body composition. The participants consisted of 802 males and 825 females, living in the city of Changsha and the neighboring areas. The genomic DNA was extracted from peripheral blood leukocytes. They genotyped each participant in accordance to a Affymetrix protocol. The Structure 2.2 program was utilized to find any possible population stratification in the research sample. Participants were clustered by randomly selecting 2,000 SNPs at the genome. The CNVs were identified using Canary algorithm, which is implemented in BAT Switch software. 7 sites of lean mass were analyzed: left and right legs, left and right arms, trunk, and the whole body. They adjusted each phenotype by age, gender, and the first 2 principal components were calculated from 2,000 selected SNPs. They normalized residual phenotypes by inverse quantile of standard normal distribution on analyzed phenotypes. The association of lean mass and CNV were performed by a linear regression model using Plink and used CNV as a predictor of lean mass. The main characteristics of the sample are summarized in this table. One CNV2073, was the genome-wide significant with p-value of 6 13.22E/7 for lean mass at right arm. Figure 1 shows the Manhattan plot of genome-wide scan for phenotype. The affiliated of CNV and lean body mass is apparent in many other locations. However, none attain a genome-wide significant level. CNV 2073 locates a chromosome 15, Q13.3. Compared to 2 subjects with 3 copies, they had 6.7% lower mean lean mass on the right arm. Participants with 4 copies had 11.2% lean mass on the right arm, as you can see in figure 2. Gremlin 1 locate in the region 15q13.3, which is covered by CNV 2073, is a candidate gene for lean body mass, which is reported by genetic linkage study Zhao et al. 2007 and molecular function study Severance et al. 2009. In conclusion, the authors have conducted GWAS between CNVs and lean mass in the Chinese population, and they found significant gene Gremlin 1 in the region 15q13.3. This study contributes to the understanding of the genetic determinant underlying sarcopenia-related phenotypes and constitutes to studies as a whole.